This is theme for Monday, May 18th meeting. And I think I want to get started. There's a lot going on with the research. So I want to, before we go into a report of how things are going at the hospital, I think I'd rather turn it over to Saurabh so he can give us a brief summary of the FDA submissions that we're planning to do in the next week or two, because we need some help with some local PI, which mainly would mean residents at some of the hospitals. So maybe before that, Frankie, why don't you talk about the plasma project and what you need at Palm Springs, and give everybody an update.
Sure. So far, we've enrolled 4 patients into the plasma trial. Right now, they're all at Larkin, Maine, South Miami. So now we're starting to get the systems in place, figure out how we're gonna get the consent, get the randomization. So right now our two big one of the big changes is we increased the length of time for enrollment to within 48 hours of hospitalization, so that we'll have more patients who are eligible, and we're a little bit less reliant on a quick turnaround from the lab. It gives us a little bit more wiggle room with that. So our two big projects now moving forward are going to be getting some help trying to enroll people at both campuses, identifying patients that are eligible, talking to the primary physicians to ask, is it okay that we enroll this patient in your trial? And as well as starting to collect the data from the patients that we already have.
So your issue right now is that you need someone on the ground at Palm Springs. That's what you're missing right now.
Yes, pretty much like a clinical trial coordinator recruiter role to identify these patients as they're coming in.
So Maria, is there a resident or Manny? Is there like a senior resident there that can play this role for plasma?
So right now we actually have no one. So I spoke with Catherine this morning, and we have 127. We have no available free. Every resident is in the hospital at some capacity or another. So we actually have rheumatology residents coming in to help. That's how big our census is right now.
Yeah. So we would need basically someone that can identify who's a candidate for plasma. That's basically what we need. I don't know, Catherine, would your QI people be able to do that, or?
I can talk to Maria, and I can talk to PI and see. Like Manny said, we have 73 positives right now in the hospital. So everybody's stretched out, but let me find out if maybe somebody from PI, if they can at least identify.
Or you know what? Do we have medical students, Manny, that we can assign this to? I know there's Chris Johnson. I know Frank. You had a few medical students assigned to your team, right? Wasn't Chris Johnson from? I think he's from LECOM.
Jack,
yes.
We just took on AmeriClerkships. I think there's another student coming in from AmeriClerkships. Maybe you can use them.
We have 5 on the IM service right now, so we could do that.
Can you lend at least one to Frank so that he can, because without that, that's why you don't have any, because you don't have anyone looking for them.
Yeah, absolutely. I can speak with Frank as soon as we finish it. We'll set it up.
I mean, you should probably have 3 or 4 by now, or
yeah,
like Larkin South does. So let's get that going. So Manny and Frank can communicate. Okay, so Saurabh, do you want to start with whatever? Just give everybody a very quick summary and what you need so that we can just get things going.
Sounds
go
great.
ahead.
Yes, sir. So effectively, we are looking to submit a clinical trial investigational new drug proposal for BCG vaccine on the basis of its use in non-TB viral conditions previously. And we believe that it has efficacy through helper T1 cell mechanisms, as well as actions on innate immunity, such as innate lymphoid cell activity boosted. So what we proposed is to write a IND, and we have been successful in completing a IND write-up, and it is ready to submit. However, we would need a private and we need a principal investigator. So we hope that we can work with one of the respected residents, fellows, or attendings at Larkin so that they can take the lead on this trial. We hope to learn from you. Pardon? Yeah, we hope to learn from you and do our best to make this project a success. And we will be having another second trial on BCG as well. So the first trial on BCG effectively just looks at BCG in a simple non-recombinant vaccine form. We are in particular doing a intradermal 0.1 unit dosage, and that's what's done in most countries. And this will be done in order to compare BCG amongst the control groups. So we will be using a control group of chelated zinc bisglycinate 3 times TID, and we'll couple that with a zinc ionophore to understand what exactly BCG does in comparison to a viral entry inhibitor such as zinc with zinc ionophores. And then we will also stratify the enrolled BCG naive patients by age. We will have population ranging from pediatric to elderly. We will randomize the patients into 3 categories by block permutation: those receiving a non-recombinant BCG vaccine, and those receiving the chelated zinc bisglycinate TID with the zinc ionophore, and those receiving saline. We will also have 2 retrospective groups, one who received the BCG before 15 years prior to current day, and also those who received the BCG between the onset of this trial and 15 years ago. So we look forward to working with you on this. And then we will also have a second trial for BCG. In that trial, we will focus on a intranasal recombinant dosage administered to the enrolled patients in the trial. We hope to also look at the IgA in particular because of the mucosal immunity which is stimulated by BCG, which is given intranasally. So we would like to further investigate biomarker elevations in mild, moderate, and severe COVID patients. If enough patients will enroll in the trial, then depending on whether the other centers' IRBs approve our trial, then we can also have enough patients to look at particular biomarkers between the sicker patients, between the ventilated and non-ventilated, between the DIC and non-DIC. And this will allow us to exactly quantitate the biomarker values, which will help us be able to prognosticate better approaches for future patients. We will be able to identify what exactly happens on a transcriptomic scale when BCG is given. So I think this is going to be a very successful approach, and we will also need a PI for this. So we look forward to working with you, and I hope to learn a lot from you. We also have a couple trials going on quercetin. We are going to be looking at doing an open label clinical trial
So, sorry, let's stop with that one, and then we'll go to quercetin next. So
Sure.
for the BCG, for the first phase of the trial, we need local Larkin residents at both sites to be PIs. Hopefully that have completed the CITI course. Are there any currently listening that are willing to do that? Manny, can you find us people that are willing to serve as PIs?
Yeah, I can ask some people today. I'll speak with Alina, see if we can find someone on either one of our teams to do that.
Right, and then obviously you always had the program directors like Dr. Fontella and Dr. Abreu, but we do need kind of a resident on site at each facility. And for South Miami, Humberto, can we get someone from there for BCG?
Yeah, we're looking at someone that is willing to stay active.
So it will be, George as the program director will be involved, but we need, and we need them to take the CITI course also so we can list them.
Yeah, Dr. Michelle is here, is listening, and we'll look at someone.
Okay, so that's BCG, and so the next one is go ahead, Saurabh.
Sure. So I also want to let Dr. Patel mention a little bit about the recombinant vaccine approach we will take with the second trial. He can provide some more input on that. And here you go. He is ready. He's on the call.
Yeah, I see you, Dr. Patel. You seem very busy. Are you in India or here?
No, I'm actually in Chicago. So let me just give
Chicago,
a background.
that's right. You said that before. I should have remembered.
Yes, Dr. Michelle, and some of us, we already spoken before in the previous meeting. So my background is I'm an IMG. I'm a tutor for USMLE Step 1 to Step 3. So I've been reading a lot of, immunological component of how BCG is generally, I've been reading a lot. So how BCG is going to affect our entire primary defense mechanism. It's some epigenetic, transcriptional, and metabolic changes which occurs independently of adaptive immune system. So even patients with SCID who don't have B and T lymphocytes even functioning, they can even have boost up their immunity when it comes to primary. So I actually told Saurabh, thanks Saurabh for bringing out the plan. Intranasal approach, because when we think about for normal healthy patient, if we inject that intranasally, or we can, any aerosolized form, that way of, recognizing patterns which will be improved with BCG alone. Now there is a certain trials have done there in past with SARS-CoV-1. Now, Jaran, what data has said, I know this is like an entire flow that I have created, and I know it will take like 5-10 minutes, but it's just the entire thing of like how to I need a lot of mentorship for this, and I think this is a thank you, Dr. Michelle, for allowing this platform to have some mentor where I actually learn too. So the main mechanism is do biogenetical modifications of certain epitopes of current coronavirus with BCG, because we are already doing a BCG trial, what Saurabh generally right now that we are going to apply, the first clinical trial. With that idea, we will have some genetical informations of these macrophage, natural killer cells, dendritic cells as a primary host defense. So their conformational changes, we also have to do another study. So there are 2 to 3 BCG clinical trials are also underway in different topics. So the first one will be combined vaccination that I'm talking about is with epitopes of current coronavirus. It could be the spike protein or any envelope protein which is similar, or past coronavirus which has the very genetical information same, like SARS, 70 to 80% information is the same. So that epitopes can be biogenetically engineered with BCG. So it gives a dual immunities, because BCG has been anyway has been the best option. There was no trial showing a successful tuberculosis transmission. Even if you had BCG, people also got tuberculosis. So people look at it as a futile flop plan. But what happened, host defense mechanism actually got resisted with the very lethal microbes like respiratory syncytial viruses, herpes. All of these viruses, less frequently were found in BCG vaccinated. So that is a memory-like response by primary host. So we are planning to biogenetically engineer coronavirus either past MERS or SARS epitope, or if possible, we need the current epitope. If it is possible, they have done. Wuhan has February 11th, 2020. They had a study done of entire genetical sequence. And I think it is available for any scientist or any clinical trial to run in a longer, if there is a successful trial for this coronavirus. Now, we are only suffering for first wave. There'll be another wave could also come. In the virus history, it has been always, there is a second wave. So what we need is generally two things has to be done. Host defense has to be improved, matured. And the second one should be decrease viral entry. That's generally as a prophylaxis.
Okay. So who's interested in being the local PI for BCG then? Do we have, I guess, Manny, you'll work with somebody there, and Saeed on the call? Actually, either Pedram or Humberto. Can you figure somebody for this?
The thing is, we sorry, Dr. Michael, we just need to find some companies who just can help creating this, because this could be a game-changing, clinical trial, which has been done.
I think Kinal's great at getting companies, so she already got Sofluza's.
Oh, okay.
She'll help you with that piece. But I mean, as far as a local PI, I think Mendez is on the line. He's our infectious disease director. Maybe he can get involved. But Humberto, can you work it out then with? Do you guys have Saurabh's information?
Yeah, I do. I have his information.
Okay, so let's just make sure, because we have all these people working very hard on doing this. This past weekend, I got like almost 20 certificates from CITI for training for research trials from this group. So they're working day and night. So I mean, I'd like to have some local support from the local Larkin residents for them, so that we can at least identify who can serve as the PI locally, as we're about to submit these to the FDA. Okay, so let's move on to the next subject, Sora.
Yes, of course. So the next thing we will submit after BCG 1 and 2 is quercetin. So Jordan is on the line, and I just discussed with him about some novel aspects about quercetin and its metabolites and their interaction with something like pregnon X receptor and how that will affect protease inhibitors. So he will tell more about that.
Okay, so the working title that we have for this study is an open label clinical trial, blinded clinical trial on natural quercetin versus synthetic protease inhibitor, such as lopinavir, as a prophylactic treatment against COVID-19. The reason why we want to go with quercetin or investigate quercetin and its effectiveness against coronavirus is because in the past it's been shown to have antiviral, immune modulating, and anti-thrombotic effects, in particular in association with coronavirus. Specifically, there are studies that have shown 3CL protease inhibition. Now, the SARS, this is from the original SARS from the early 2000s. Investigations showed that there are two major proteases contained within the virus, 3CL Pro and PL Pro. 3CL Pro being the more important of the two, as it cleaves at 11 sites, where the PL Pro only cleaves at 4 sites. That's important because the cleavage of the viral polypeptide is integral for the propagation of the virus, and so quercetin has been found to inhibit the 3CL Pro protease thus blocking 11 cleavage opportunities for the viral polypeptide, and hampering the auto cleavage of the polypeptide and hampering the propagation of the coronavirus. Again, that was with the original SARS. Now, there's another point where that I'd like to make, where quercetin has immune modulating effects. Specifically, it has inhibition of LPS toxins, as well as it inhibits LPS toxins, and thus decreases the amount of TNF alpha, IL-1 alpha, IL-8.
What about if it comes back negative and he's no longer in isolation?
Sorry, can you repeat that?
No, that wasn't intended for you. There was somebody didn't mute their mic. Go
Oh, okay. As well as so beyond the inhibition of LPS toxin induced effects, there's also an inhibition of inflammation from COX and lipoxygenase. So it has a NSAID-like effect on the body in reducing inflammation, as well as some inhibition of histamine, tryptase, and pro-inflammatory cytokines that has been found. Quercetin also has immune modulating effects on Th1 cells. It has an inhibitory effect on IFN gamma. It has a stimulatory and inducing effect on IFN gamma and Th1 cells, thus inhibiting Th2 derived cytokines and secretion of IL-4. And in addition to that, it has immune enhancing. These effects have been shown to exist also in vivo studies. In human studies, there haven't been as secure of results, but in animal studies and animal in vivo studies and in mice, it's been shown to hold up. Okay, so the next point of using quercetin instead of a lopinavir would be that its metabolites, the metabolites of quercetin, have shown antithrombotic effects, which is specifically important because of the hypercoagulable state and severe inflammatory state that has been found in association with the COVID-19 virus. One of the major questions I've heard from people, from just people that are concerned and listening to the news, is the strokes that have been happening and the embolic events that have been happening. So that would address that in part. In addition, there's two specific metabolites, the isoramnitin and the tamarixetin, that are the specifically antithrombotic metabolites. And when we're using isoquercetin or quercetin, metabolism within the body has it switching back and forth between all of these metabolites. So if you give them isoquercetin and then test their blood levels, you're going to see elevated levels of the antithrombotic metabolites of quercetin as well. And then the fourth major point that I wanted to make about quercetin and being used specifically against coronavirus is that there's a study using a recombinant virus with HIV and SARS that showed that showed inhibition of viral entry into host cells with a EC50 of 83.4 and a CC50 of 3.32. Okay, so specifically, I mentioned that we wanted to use isoquercetin, otherwise known as quercetin 3 glucoside, instead of just a plain quercetin. That has to do with bioavailability. The isoquercetin is, because it's attached to the glucoside, it is carried across the intestinal cellular membranes by the SGLT transport, thus has more efficient absorption in the gut and increased bioavailability. And again, once it's absorbed, it has the ability to transition between the various metabolites of quercetin. So another thing that Saurabh had wanted me to mention was the excretion of quercetin and isoquercetin. They're all water soluble. Isoquercetin is especially water soluble and gets excreted by the kidney. It has some metabolic interactions with CYP enzymes, specifically the the metabolite. Okay. Tamarixetin, sorry. Specifically, the metabolite tamarixetin has interactions with cytochrome 1A2, 3A5, 3A, and 3A4, while the metabolite isoramnitin, I'm sorry, these are all mouthful for me, has specific activity with the 3A4, but not the 3A5 cytochrome enzymes. I was speaking with him about how this could be a problem, or not a problem, but how we need to consider this with patients, especially the elderly population who may have heart failure and be on several medications that interact with the cytochrome enzymes such as amiodarone and whatnot, and how this is going to affect their prognosis, being one of the most at-risk populations in association with coronavirus right now. He had mentioned the pregnane X receptor polymorphisms and how they affect concentrations of protease inhibitors, which are involved against activity of the coronavirus, suggesting somewhere down the line there may be a good opportunity to see if its quercetin is more effective when given in combination with a protease inhibitor. Beyond that, the study population that we wanted, or the study, where did it go? Okay, the groups that we wanted to study, we wanted to have a prophylactic group for people who test COVID negative, and we were there's a similar study that we're basing ours off of, in which their prophylactic group uses 500 milligrams of the quercetin daily, whereas the treatment group uses 1,000 milligrams of quercetin daily, or takes 1,000 milligrams of quercetin daily. We wanted to up that. We wanted to look into doing a weight-based dosage, and we wanted to try giving 1,000 milligrams prophylactically, and then we have calculations yet to be done regarding what we're going to do for the treatment group. We also wanted to compare the treatment group to lopinavir, a viral protease inhibitor. And so we also wanted to have a treatment group that was going to be blindly given, well, the treatment groups are going to be blinded, and one will be taking quercetin and one will be taking lopinavir. Then we'll also, of course, have a control of patients who have no history of COVID-19 and they are not given quercetin, in order to compare them with the prophylactic group. Beyond that, was there anything that you wanted me to mention that I missed, Saurabh?
I just want to say that if the quercetin metabolite is present in the system, the duration of action will be longer, and it'll also interact with the pregnone X receptor in a way that can possibly lead to synergistic action once combined with any navier protease inhibitor. It's been shown to interact with atazanavir, and we will work with lopinavir and any other protease inhibitor which may be up and coming based on EC50 concentrations. So we will look to do a control group, which, an experimental group, which has a combination effect of the protease inhibitor and the natural quercetin with its metabolites. That way we can have possible synergistic action and evaluate that.
So, sir, so we have two groups potentially that we can test this with. One would be individuals that live in assisted living facilities or nursing homes. Mm-hmm. And the second group could be our own employees, our residents and our staff. Okay. We're thinking of offering this as a preventive cocktail, kind of. So this could be part of the study. So in total, we're probably talking about 1,500 or so employees. And then in the ALFs, it's a large number of ALFs that we would, so there are different populations, obviously.
Yes.
And we can follow different parameters. But just so you know, I think those are the two groups who could possibly test.
Yes, sir. I think that's a great approach.
Okay. So is that all for quercetin? Is there anything else?
That's it, sir.
Okay. So next is, what's next?
Next is plasmapheresis. So okay. We will be looking to work with we're looking to create 2 trials on plasmapheresis, and we need co-PIs. So I hope people are interested. So with plasmapheresis, we as also known as PECs, we will create one trial in combination with Medrol, because we are considering the cytokine storm involvement. So we have to assess whether it's appropriate to give plasmapheresis individually or with Medrol. And we're going to do this amongst the severe COVID patient population because inhibition of inflammatory activity is beneficial when the patient has reached a condition of cytokine storm. Otherwise, the inflammation is necessary and vital actually to allow the innate lymphoid cells and NK cells to inhibit the viral activity naturally by the body's compensation. So once someone hits cytokine storm, they're effectively decompensated. And the way I see it in my mind as an analogy is kind of like the heart failure. You have one approach for compensated heart failure, and then once the patient decompensates, you have to use different drugs. So the goal here is to evaluate where is that line exactly? How do we specifically identify that cutoff point where we go from compensated normal inflammation to decompensated cytokine storm? And that way we will know exactly when to suppress the cytokines, when and how to do so. Another study which we are interested in doing, which is along the same lines, is with tocilizumab. Tocilizumab, as you know, is an interleukin-6 blocker. It will work to decrease acute phase reactant involvement and also prevent the onset of hypercoagulability. So we can take an approach to compare tocilizumab involvement against Medrol. That will allow us to assess whether we need complete blockage of the inflammatory pathway or whether just IL-6 is enough. Because IL-6 has been shown to be key in involvement with many studies, many clinical trials of COVID. So we can use this approach of immuno and inflammatory suppression for two different trials here. One is plasmapheresis compared with Medrol, and the other is Medrol compared with tocilizumab. And both these approaches have been shown to be highly popular in terms of approval because there are quite a few clinical trials which have involved these angles. So the benefit of this is that once we use a company called Olink to assess the protein biomarkers involved, we will be able to take a transcriptomics approach to identify the exact cutoff point at which we need to give Medrol or tocilizumab or entire plasmapheresis. And that'll exactly lead us to better prognostic care.
So when you say plasmapheresis, I guess we have equipment for that at the hospital, right, Sandy? We don't need that. It's not photopheresis, plasmapheresis.
We do not have a plasmapheresis machine.
It's only photopheresis?
It's photopheresis.
Okay, so we need to look at who would do that, a dialysis company?
They have the ability to do that. We have a contract that includes that, plasmapheresis. I would have to check the contract to see how much they charge. But
Check up
we
both
do have a
sides because
contract that
we
is
need to
included.
figure out as far as cost, so we know what the cost is going to be. So let's check that so that when Saurabh is more advanced with this, we can already have a budget for it.
Okay, I will.
Okay, and then Saurabh, let's keep
Yes,
going.
sir. Sure. So other approaches and angles we were taking with these IND submissions include, as I said, tocilizumab, and we have also already mentioned quercetin. Quercetin, as we talked about, will have one trial with quercetin versus lopinavir. The other trial will work on prophylaxis, whereas one will work on treatment. And then we can also take a biomarker approach with the solely plasmapheresis trial, because we will have one trial of therapeutic plasmapheresis in mild, moderate, and severe COVID patients without any comparison to any drug, just to evaluate what exactly happens to these biomarkers when we subject patients to plasmapheresis, what is going on in terms of a biochemical scale. And that'll also help us improve care. Further studies can involve EGCG, which has a very low EC50 against viral replication and activity. EGCG is a green tea extract catechinin, and it has been shown to work as an approach which is multidimensional, similar to quercetin. So we can take a control group, an experimental group, and apply EGCG as an administrative dosage, and we can evaluate the right dosage. We can also do a comparative trial of multiple dosages to see what is best for these patients, which trial will reduce the ventilation, which trial will reduce the stays in the hospital. We will look at these kind of outcomes, and that will let us take the angle at which we can get the right dosage of EGCG, because it's been shown to be effective as a antiviral, and it's cost effective. It's much cheaper than tocilizumab, so it is definitely worth investigating.
Okay.
Sorry, Dr. Misha.
Yeah, go ahead.
We also discussed in last meeting that we'll also compare quercetin with astaxanthin, because there was a recent new study was done which was preventing viral entry. Astaxanthin is a highly antioxidant component that generally cold water fish or krill, they generally eat those algae and phytoplanktons. So the red pigment in salmon, that is astaxanthins, that generally gives that pigmentation. So I think with quercetin has a similar effect with astaxanthins, but astaxanthins efficacy was shown better in recent study. Maybe we should also compare that two group.
Yeah, I think we can definitely do that. We can evaluate different dosages of astaxanthin as well. I believe astaxanthins don't have a high risk of side effects, so this particular family of compounds will have a high therapeutic index, so it'll be a safe experimental or control group, and we would not have too many adverse effects to worry about.
Yep. And generally this kind of treatment would take only 2 to 3 months, so the duration of the studies are also not long.
Right, I think it's yeah, I think we can focus on approaches which have quick trials. That way we get quick results and make an impact.
Yep, I agree.
Perfect.
So to run that project, we also need a PI.
So for the chief residents who are listening, Humberto, Saeed, Manny, John, and Alina, we need to kind of write, we need to start coming up with either residents or attendings that are going to be willing to be listed as PI or co-PI. But we could discuss that offline. It could be any attending at any of the hospitals that is interested in a particular study. So actually, Dr. Mora is on the line, so maybe he can also talk to some of the attendings there. Obviously, the program directors are going to be automatically listed, but we need someone else to also be kind of the champion at each site for each of these programs. Sarab, let's talk about whatever we haven't discussed so far. I don't think we talked about ozone yet, and
Right.
Go ahead.
Yeah, sure. So we're also working with Xofluza and ozone, and I'll be working with other students who are leading these projects, and I will be helping to guide them in their approach to the IND submission. Xofluza is on hand at Larkin, so we can do a study with different dosages of Xofluza. We can create a comparative control group as well, and we can also assess with a mechanistic drug which is commonly used in standard of care, such as Medrol, to create a control group. And then we can also evaluate the biomarker elevations with Xofluza and this will be a study up and coming, which we requested PI for. Also, we would be working, I'll be working to show the other students how to do an IND for ozone. And with ozone, the trial will be led by Dr. Richter, and he has about 20 years of experience with ozone, and he has a protocol that he developed, and that'll be going forward as well. And anyone can feel free to work with them on that.
So let's turn it over to, I guess, Kinal, are you heading the...
Yeah, Kinal and Sanjay will be leading the projects for Sopusa and ozone respectively. They can tell you more about it.
Yeah, so Kinal, give it a summary of our talk with Genentech and how that's going, and then we'll turn it over to you. Go ahead.
Sure. So we had a call with manufacturers of Xofluza, Genentech. They're quite encouraged to hear that some of the stories that one of our investigators had, or physicians had shared with them. We're waiting to hear back from them. I believe we had Dr. Marcos and Dr. Michelle on that call. So Dr. Marcos and Dr. Michelle, as well as a couple of residents I'm working with, we will gather the data that we have so far of both outpatient as well as some of the inpatient who were on Xofluza. As of right now, we are thinking about designing at least one retrospective observational trial just to gather the data we have. And we're also thinking about coming up with a prospective single arm observation study on Xofluza. And I believe to my understanding, majority of these patients were on Xofluza 40 or 80 milligrams based on their weight and age. And I believe they were also given azithromycin 500 milligram in addition to the Larkin protocol of vitamin C and zinc, as well as famotidine. But again, we need to kind of gather this data to figure out what the consistency is. So I'm looking at it as two different things. We want to analyze the data that's been collected previously. And if we design a trial, it would be trial moving forward. So the role of the Genentech here would be to provide us with the medication, and it would be our own trials. That way, these medications, I believe patients are having to pay $200 out of pocket if they can. So Genentech, if they got involved, the cost for the medication would be covered for patients.
Thank you, Kinal. Dr. Richard, do you want to talk about ozone, or sorry, your person that was in charge of ozone is also here, or?
So I placed Sanjay in charge of ozone because he expressed the most interest, and I'm not sure if he's on this meeting. He's actually in Nepal, so he might not be.
Oh yeah, he might be sleeping now.
Yeah, I think so.
Dr. You've been in touch with Sanjay,
yeah,
right?
I was contacted by Sanjay, so we were.
Talk a little bit about how the IND is coming along for the submission for the FDA.
Well, we have already kind of I reviewed one of the protocols you sent me. I put some observations, so I guess we just need to kind of adequate to what we're gonna do here in the United States, because this protocol is based basically on equipment and bags that they have in Spain and Italy. So once we just adapt to the US protocol, yes, I mean, that's basically, it's very similar. So I would say it's almost ready. That we're just waiting for the right equipment to arrive, the transfusion sets. And I spoke with the nurses. I guess there were 2 nurses selected in Larkin South that we're gonna be working on Wednesday. Maybe
Wednesday at 2, Doc.
Yes, exactly.
Wednesday at 2 o'clock. Yeah.
At 2 o'clock. So hopefully after that, if there is a patient, there is one I know in ICU that they already
ICU.
requested, yes, they requested a consultation. So I'm gonna review his chart, and that may be one possible first candidate. Well, I'm still working on the informed consent. An email Alina also this morning, see if she adapted something to Larkin's protocols. I gave him an updated informed consent. So maybe just one, since I'm reviewing the city guidelines now, there is maybe only one more point I want to add to that consent, which is not in there. But yeah, that's where we are right now.
Okay, and what about the tubing Mercedes? Has that been ordered?
Yes, the tubing is already arrived, Doc, and it's already been reviewed by Dr. Nick. It's with the exact one that Dr. Richard ordered.
Okay.
And is this going to happen at Palm Springs too, Maria, or what's going on over there?
We want to get it started here, but we also need to schedule for the training for our site as well.
Okay, so you work with Dr. Richter on that.
Okay.
France,
are you available for that?
Yeah, sure. We haven't coordinated anything yet because I was waiting for the equipment to once we try the new equipment, we are ready to go everywhere. I mean, the training is just
the ozone concentrators, Cindy and Catherine. We were going to order one ozone concentrator per hospital.
We ordered it. Let me find out where we are. I know we looked at it about 2 weeks ago. So I'll find out if it was already.
I have two equipments. I can, temporary put one in each hospital. It's not a problem.
Richard will lend us his temporarily, but we should order our own. It's only $2,000 or something.
Yeah, we ordered it down here as well, Jack. We just don't know when it's arriving. I'm actually walking to Harry's office right now, but the doctor had mentioned he would lend us his in the interim.
Okay, we want to make all these options available.
The ozone.
Yes, sir.
I'm sorry, Dr. Richard, do you have anything else?
No, that's good.
Saurabh, what else are we missing?
Other than that, I believe that the next step would involve taking a molecular-based approach, which will allow us to identify oncoming targets. And I believe that our strategy with biomarkers, with transcriptomics, will let us have a snowball effect, which will lead to identification of the best, most versatile markers. And they will let us find the exact points at which COVID patients decompensate. And then we can tailor our treatment approaches accordingly.
So we had a lively conversation earlier today with Dr. Felix from Larkin University, and then also from the people that are running the molecular lab. We are able to do the whole sequencing ourselves internally because we have the equipment to do it. But there was some debate about how it could be structured. So I left it kind of to that team to get together. Arpit, Aaron, I think Madhu, you were involved too, and Dr. Felix, and maybe Dr. Sanchez Gonzalez, so that we can work out some of the details. Is there anything else we left out, Saurabh?
I believe that's all, sir.
Okay, so other than that, I was approached by someone that wants to put together an FDA trial for photophoresis, and then the difference is that we gave up on it last time because we do have the equipment, but we don't have the kit. The kit costs like $2,000 for just to have per treatment. So we decided that it was just too expensive. So we found this person that actually developed their own photophoresis machine, and they want to, I guess, eventually sell that. So they said they've already tested with other viruses. So that's another trial that's coming up soon. I'm getting the protocol later today, and then I'm going to put it, Saurabh, I'm going to put it on your Telegram group. So then
Sounds great, sir.
If you got anybody that's not doing anything right now that wants to sign up, and then we'll start working on the IND for that. And then finally, there's one group that is doing some stem cell treatments They did a couple of Baptists, and apparently the patients got better. So they want to expand to our site. So that's also, they're providing all the stem cells and everything else. So it'll be just kind of like a late trial for us, for people that are already in ICU and as a last resort. I think that's pretty much it. I think if Saurabh, do you know have anything else?
And that's all. I've received some contacts from Bina, Dr. Rad, and Dr. Zadeh, and also Dr. Briones. So I believe that I can introduce them to the BCG Telegram groups and also the quercetin groups, and they can help in teaching me and everyone how to what the best approach is for our next steps.
Perfect. Kinal, do you have anything? Madhu, anything? You're muted, Madhu.
Thank you, Dr. Michel. I think it's a great idea. Now you discuss about the stem cells. Maybe myeloid stem cells maturations, which occurs after our BCG patients, those can be used towards somewhere in different era.
So we'll talk to those. It's actually pretty approachable. So we'll talk to them once that's going. So that sounds good. Okay. I want to turn it over to Humberto to give us an update on what the outcomes have been so far with the new protocol. We haven't changed the protocol, right? We're still doing the same things, the Xofluza and the plasma.
Yes, everything has been the same. We have noticed that in the complicated patients, the use of mucomis has been improved and have reduced the timing of the ventilators. So those findings have been proposed for Dr. Yaskan, and one of the residents that is rotating over there always noticed. We have a Dr. Preet that has been sub-I, has been rotating with us, and now he's very interested in this new trial. I'm going to introduce you, and he's going to talk to you about this new trial that he's developing.
Okay.
Okay, Preet.
Hey guys, thank you for having me here, Dr. Michel, and everybody else. So basically, what I looked at the medical literature regarding the COVID, and what I found out is that there is some biochemistry, molecular basis for what we're seeing. Basically, there's the glutathione component, right? And N-acetylcysteine, which is mucomist, it's inhalational mucomist N-acetylcysteine. And what it does is it becomes glutathione, and glutathione is a reducing agent. So when we have sepsis from anything, could be COVID, could be bacterial pneumonia, this glutathione gets depleted, and then that's say that this glutathione inhalation or IV can translate into clinical improvements. There are a lot of very impressive trials going on right now, but if you notice, there's like a lag between the clinical effects. So what I'm saying is maybe mucomist along with you can even do IV glutathione. These can translate into clinical effects or clinical improvement where the patient will feel less short of breath, things of those nature. And there's a lot of literature. Glutathione, so basically it has two effects, which are very important for this COVID multifocal pneumonia. Its first effect, so what it does is it breaks the disulfide bonds. And these bonds are found in the mucus. I was reading some articles based on the autopsies. I believe that it was New Orleans. And what they found out was in the cut section, they found a lot of edema, a lot of hemorrhage. So mucomist, it might just, once we administer it, it might just give the patient a lot of clinical improvement. And as far as evidence over here at Lark and South, we tried it, and it's really impressive. The O2 saturation, it went up pretty quickly, and Another important thing is that glutathione can only be found via the HMP shunt. And HMP shunt obviously is related to the G6PD. And if you notice carefully, the G6PD has, or some of the patients that we see, they have hemolysis going on. They have the typical findings of intravascular hemolysis. And so it's not a bad idea. The scope of NAC, which is N-acetylcysteine, and the glutathione, it could be pretty big. And I mean, we're focusing on all these very impressive treatments. You know, they're expensive also. But what about something as simple as N-acetylcysteine? So it has two effects. The one, it breaks the disulfide bonds, which is in the lungs, as well as the von Willebrand factor. In all these intravascular hemolysis that we're saying, where there's platelet consumption going on, that's a particular area of target. I believe, you know, it is something that we should work on. I'm just looking for a little bit of guidance, you know, as far as like how we're gonna set up the trial.
Why don't we set it up as a Saurabh, can you help him with that?
Yeah, definitely. I'm very familiar with N-acetylcysteine and its properties as an antioxidant, and I'm very familiar. I've actually done research on glutathione before at the Cleveland Clinic, so I can definitely assist with that. And I believe that I did see a clinical trial with N-acetylcysteine, so it's a feasible approach, and we can perhaps compare it to, we can do control groups, I think, with standard of care, and then have an addition and experimental group of different dosages of N-acetylcysteine to identify the best dosage. And then we can incorporate healthcare workers, pediatric populations, elderly. We can stratify the patients in different age groups. And we can also have an experimental group with similar antioxidants that have mechanisms which allow for raised glutathione production, because there are other compounds available that will significantly raise glutathione.
Right.
So, Saurabh, do you want to post your contact information on the chat, and then maybe you guys can connect, and maybe let's create a Telegram group for this, and then we'll add any other. How many do you have in your group that are not assigned to a project right now, Saurabh? Because you have like 90
in my
people.
group that are not assigned to a project, you mean in Team 11?
Yeah.
In Team 11, there are about 55 people who still need to get onto a project. So I think that there's plenty of room for more projects.
Some of them can work with him and help him put it together.
Right, right. And my contact information is on the chat.
Mm-hmm. All right, thank you.
No problem.
Yes,
So you guys will connect then. Perfect. Thank you so much. I'm excited. You guys are going to these projects. It's getting confusing, but I'm sure we'll work it out. It's great. I'm very encouraged by what's going on. I mean, over the weekend I got 20 CITI, which is research certificates from this Team 11. They worked all weekend long, just getting the certificates done. So we're in pretty good shape. Humberto, do you have any more updates from South Miami?
No, we have, like we were saying, 32 patients COVID. Only we have one intubated. It's only one intubated, right? Yeah, only one intubated, and the rest of the patients and evolution is well with the protocol that we already have. We are working one of the group we're doing the remdesivir. One of the other group we're treating with the early plasma, and we follow the general protocol that we already established. So,
how many doses of remdesivir have you received from the state?
Actually, we have well, yeah, but how many patients? Like 4 or 5 patients. So we have 4 or 5 patients. All the patients are receiving the first dose is 200 milligrams, and we continue 500 milligrams until 5 days. So far, the results, there is no difference within 5 of them.
So you have no issue getting the remdesivir right now?
We're receiving the medication very quick.
Okay, and what about Palm Springs? Are you guys getting the remdesivir from the state, Alina? Or
Can you hear me, Dr. Michelle?
We are.
Yeah, go ahead, Jeffrey.
Yeah, so right now, ICU, we have 2 patients already on remdesivir on 2nd day, and we further request 3 more, 3 other patients. So going to be total of 5 patients in-house that is going to be on remdesivir.
So you're getting it easy. It's not, they're not giving you a hard time.
Over the weekend, it's difficult because I think the delivery part, the transportation is the part that delayed the process over the weekend. Otherwise, everything was smooth.
And Jeffrey, how are the patients doing in Palm Springs? How many, are we having deaths? Are we having?
I think all the biomarker actually trending start to improve with the marker. However, I think it's the collateral comorbidity actually start to still play the role that the reason why patients not having drastic improvement. So yeah, still monitoring.
Okay. Alina, anything?
No.
Manny, anything?
No, I was just gonna say the same thing. I think in the ICU, they're doing pretty well, similar to what he said. We have 73 positives, 2 vented patients. We have the one currently on the remdesivir, and we're waiting for the other ones. But otherwise, we're doing well. The hospital census is the highest I've ever seen it in 3 years of being here. So everyone is doing a fantastic job. All the nurses, staff, everyone, trying to take care of these people as much as possible. So.
Great, thank you, Manny. Tariq, do you want to tell them a little bit of you? Tariq is a resident at Kinal Regional, but he's joining all of our groups. And actually, he has a little report about how he introduced the plasma over in Kinal. Do you want to go ahead, Tariq, and tell them a little bit about it?
Yeah, sure, definitely. Thank you for giving me the chance to talk about this. Yeah, so actually, one of the things like I figured out when I was trying to introduce the convalescent plasma, basically not a lot of people knew about it. And even when I tried like to discuss about the convalescent plasma, most of them like, what is this? And they want to know like, what is the answers regarding what is convalescent plasma exactly. So the process started initially by approaching the ICU physicians. I brought to them the idea of convalescent plasma and how it's working and everything. And they started questioning me like a lot of times like about it. And they kept like asking what is it and what is the indications and all of this until everything, I showed them like evidence base behind this. So they agreed like to give this for some patients. So we started initially on 2 patients. This was the first approach. And then from 2 patients, the next day, again, the ICU physicians, they contacted me that, okay, let's give it to this patient. So we increased it to 5. So we applied like to the five of them, and they received the convalescent plasma. After that, the infectious disease, he also contacted me to administer it for one of his patients in the floors. Yeah, so I gave that to them also. And then after that, like a couple of the residents and the physicians, and most of them, they were asking me like, what is the process? How do you administer the convalescent plasma, and what is needed like to do that? So I figured that, okay, so most of them, they're gonna ask, so it's better that I build an approach on exactly how to do this. So I prepared like a document for them, which discuss about the process exactly on how to administer the convalescent plasma. And I sent it like to all the residents and the physicians in the hospital to tell them exactly about how to do this. I'm trying to find the sheet, but yeah. So I send it to them, and after that, the ED physicians initially they approached and they said like, let's do a presentation and talk about the convalescent plasma, so I can explain it like to them and to understand like the concept behind it. So after that, I prepared another document regarding the background and the significance of the convalescent plasma, and how is it effective, and even the historical approach behind it. So they like the idea of the convalescent plasma a lot, and they're surprised like they're not using it. I can show you the document, but you want me to open my screen?
Well, yeah, go ahead. Can you share?
No, how's disabled attendees?
Oh, I wonder why I did that. Let me see. Let me make you co-host, maybe. Make co-host. Okay, here we go. So just the reason I'm saying all this is because, you know, it's one thing when we change behavior or we change outcomes in your own institution, but you know, Tariq has basically shown us that, you know, the whatever we change in our institution can be also replicated elsewhere to save lives. And he went from 0 utilization, am I correct in saying that, Tariq, to now they're using plasma, and he's the plasma guy. So he created this Larkin Team 12 under a, you know, not so public identity, where he's actually trying to recruit now residents from other teaching hospitals to give them this presentation, which I'm also going to share with some of the DIOs about why they should be using. So this is sort of what I'm thinking is, as we do all these other projects, these are things that we can use to help them say, this has worked. And then when we have the literature that can show it, I mean, and Tariq did a really nice job of putting it together, explaining why plasma works. We could do the same thing with pretty much everything else we're doing and maybe change the standard of care. But this is what Tariq has, and thank you for sending it to me. I've already given it to a few hospitals, and frankly, some hospitals, and I know that Patrick didn't want to go right away with other locations, but as soon as you're ready, I have a bunch of other hospitals that would love to be part of the early phase project.
Perfect. Yeah, I think that works. Like even the studies behind it, like it shows that it actually the early use is much better to do.
Yeah.
The
interesting thing about this, just for everybody to hear, one of the things that Tariq discovered was, and I know Frankie, you and Patrick probably also knew this, was that, you know, that no one has ever really proven in a randomized trial that early plasma administration works. Like, it's been tried on Ebola, it's been tried on other things, but no one actually did a randomized trial that actually proved. And the reason was that the Ebola epidemic was kind of almost waning by the time they started doing it, and so they weren't able to finally do the study. So this is the type of study that, if you look at the convalescent plasma study, this is a study that in the next pandemic, you know, hopefully not 50 years, but maybe 100 years from now, somebody's going to quote this article and say, at Larkin, they did this plasma, and they showed that it's actually, if you give it early for whatever that virus is called, COVID-51 or whatever, hopefully not, but we can start using it early on and maybe save lives early on.
That's true. I think that's a significant point to consider, because most of the studies, like even in Africa when it started, and then the Ebola outbreak, like it waned, so they couldn't get enough data to prove its effectiveness. And even like the WHO, they were saying, because they don't have a data exactly, they were mentioning in their reports that they consider it as an empirical therapy. And if you know, like empirical therapy is basically based on experience and educated guess. So this is one of the things that is significant about the trial that you guys are going to do in the hospital, is that this is one of a kind. Like it's something still not proven, and once it's done and the results are out, it's going to show that how effective is this. And even I noticed, like from the study, from going through the process, I noticed that there is a couple of recommendations that the Ebola during the Ebola outbreak, WHO said. They mentioned that the recruitment of donors living in areas in the epidemic which has broken out can offer the added value of providing a very specific artificially acquired passive immunity. Again, is the local infectious agent, because if you know, like the COVID-19 have different different mutations and they have like different species. So so it's better that we get the convalescent plasma from the area that we are in, because the antibodies are going to be very specific against the local infectious agents that we have compared to other regions, which actually, like if you get it from other regions, sometimes it's less effective due to the strain variation. So that's why, like when they tried to do this, they found a challenge. The challenge was to do was identification, selection, and recruitment of the donors. So to do this, then they found like 2 solutions. And one of the solutions, which was actually amazing, is engagement in the community. And this is the thing that you guys are doing. So exactly, you're following the right steps on approaching this. And this is where I told them, like, one of the solutions was engagement in the community. And even in the Ebola outbreak, the WHO have dealt with key considerations regarding effective information, education, and engagement of patients that recovered from Ebola disease and the communities in which they live, to consider donation of convalescent plasma for the use in the treatment of Ebola virus. So this is where I then open the document and I show them an example of how we can engage in the community. And the example was the Larkin Hospital, regarding all the teams, and I discussed to them like all the teams on how they're approaching this to engage in the community and build an effective approach to administer convalescent plasma. And then after that was the other one was the other solution was the creation of a register or database of patients that recovered from COVID-19. And this was actually an approach that Parikh had given to me. So I summarized it and I showed like what is the way that we can do it. And I'm sure that you guys are already doing this over there. So yeah, so basically the document, like it actually should prove effective. Even this is the process exactly on how to administer the convalescent plasma, who meets the eligibility criteria, what is the pathways, how to get the consent form and all of this. But this is somehow like specified toward our hospital, like because we build a consent form in the hospital and all of this, other than the My Clinic. So yeah, it actually proved effective. Like after I educated the residents, the physicians, and most of them, the program directors, and even I met with the CEO and told them about this. And now we're in the process of building like an order set in our Meditech, which is the physician, like where you place the orders and all of this. So that the order set becomes easy. It's an information system and it's an order set at the same time. So that if someone wants to order convalescent plasma, they gain information of what is it, what is the pathways, and it becomes like even the follow-up, the consent form to be sent to the nurse, the labs, and to inform everyone. It's basically like a whole information system of what exactly to do it easily. So yeah, what Dr. Michel mentioned, that I hope that this will be incorporated in other hospitals, since from my experience, I see that not everyone knows about it and not everyone understands like how to administer convalescent plasma. So if we help like other hospitals and we show them the path to do this, this is also going to be an amazing thing to do, and to put them in all in like one umbrella, even if they need the convalescent plasma, especially that you guys have a big pool of convalescent plasma, then we can help them with giving them, donating to them convalescent plasma, and show them the process, and even exchanging information between between all of us, like to, for example, to do a ground rounds or presentations, or to know about convalescent plasma. Like, so there is a lot of ideas behind this that can be done if we just if we reach like to most hospitals around us and build in every hospital volunteers that can incorporate the system, and then we build a network between all of us, and we share information, we share resources, we share everything. So yeah, I hope this will go forward, and I'm looking forward like to, if everyone is interested to join and to help with this, then definitely let me know. And Dr. Michel, I like the fact that you shared it with other hospitals. I hope they reach out, and then we start the process. Of course.
Did you eat lunch yet?
Thank you.
So what, sir?
What do you want to eat?
That was someone. Thank you. Dr. Mendez, do you have anything to say?
No, I just want to say congratulations to the team that got us remdesivir.
Yeah.
Do we have Dr. Yaskin on the line? Anybody know?
Dr. Yatskin right now is rounding with the ICU team and Palm team at Palm Spring.
Oh, okay, got it. I think we needed him. Saurabh, did we need Dr. Yaskin for the pulmonary doctor for one of the trials?
I believe we did. We were looking to discuss his involvement with the Medrol and plasmapheresis trials, as well as tocilizumab. Basically, anything that involves measurement of ILC2 activity for immunosuppressive approaches.
So can the team reach out to him, please?
Well, I mean,
not right now, but just get him in touch with Saurabh later.
What do you want to eat it with? What do you want to eat it with?
John, we can all hear you, but how you doing, John? No, I meant
Dr. Jack.
Yes.
We have a little concern with the plasma trial and the use of remdesivir, and how could that could be like somewhat masking what we're trying to point out. We have patients in which we have used the plasma, and then we have also used remdesivir. So I mean, when you get the results, like how will you differentiate what's working or what's not working?
Yeah, I mean, honestly, it's a tough situation because you have somebody. I mean, the reason you're using the remdesivir is because they're already at the stage where George?
Yeah, I'm here.
Isn't that the criteria that they're ready? I mean, in order to use the remdesivir, they have to be in bad shape, right?
Yeah.
So you're giving plasma early. So,
right.
I mean, if they don't recover, you still have that data that maybe didn't work in some cases, or even though you gave it early. But I'm not sure that it masks anything. Why do you think it masks?
No, because I mean, sometimes what I've seen is that we're giving the plasma, and then like a day later we're giving remdesivir. So I mean that confabulates the whole. I mean, you're not even giving 24 hours for the plasma to work.
Okay, well that's an issue. Why are we doing it that way?
Well, that's one thing that I wanted to bring up. We've seen that with like 2 cases or 1 case here, and I guess it's just different people ordering things.
Yeah, so we need to get a uniform. That should be in a protocol. George, I think that should be part of the protocol. Is when do you give them remdesivir? It can't be just because it's available. It has to be. There has to be some criteria.
Right. Yeah, I think what it is that we give the plasma, and then obviously, within 24 hours, the patient may not be showing improvement yet, and then, one of the other attendings comes along, and he doesn't want to wait more because the guy still has.
Oh, he just orders it.
And they order it, right? And they say, well,
how do we? You want to talk to Patrick about that? Like, do you want to put something together on when to give remdesivir after plasma?
Yeah, I think we need to like delineate that more. Yeah. Yeah. So far, we're trying to contact Patrick first and get the approval to do because it isn't randomized. So some patients will get it, others don't get it. So we're trying to
maybe the ones that didn't get it.
That's what we were trying to do from our side. The ones that got the placebo, we were giving remdesivir quicker. Exactly. But what we're seeing is a couple of patients are getting both. Like 1 or 2 of them have gotten both.
You know what? I'm not too worried about it as long as Frankie and Patrick can deal with that in their data. I don't think it matters that much to them, but because you can always, I mean, if it's happening all the time or versus not all the time, you can probably control it somehow, right?
Right. So I would say it would make, it'll have, less of an impact as long as we're giving remdesivir to both the control and the plasma arm somewhat equally, because then that should work itself out.
Yes.
I mean, it might be worth, earmarking those patients and seeing if there's, a difference between those, but as long as it's applying equally to both arms.
but your FDA protocol says nothing about not getting anything else. It doesn't.
No, it doesn't.
So giving remdesivir is not an exclusion criteria anyway. So
No.
yeah, I mean, I hear you, George. As far as maybe you want to have some way to give the plasma some time to work before people jump to give them remdesivir.
Right.
But you know what? If my theory is correct, that remdesivir doesn't work that well, maybe it won't make any difference.
Right. Yeah, we're just trying to find a way of proving it.
Yeah, I think plasma is much better, but that's my bias. Right.
And the other thing is like Sofluza or remdesivir. Wouldn't that be like a sort of like the same pathway there? Or?
I think at some point there's going to be a crunch on the remdesivir. I don't think you're going to be able to get it as much.
Right.
So at some point you're just not going to be able to get it. And when you have certain numbers, you're going to have enough people that didn't get the remdesivir, they got the Sofluza or vice versa.
Okay.
So I'm not so sure that it's going to make a big difference. But I think, for the people running each trial, if you're running the plasma trial, you should make sure that you delineate what your exclusion criteria are going to be. And the same thing with Sofluza and the others. We need to obviously keep in mind that these people are getting everything. So do they get Sofluza, ultraviolet light, ozone, and plasma and remdesivir? And then we're going to figure out what the hell worked. I don't think that's going to happen. I think there's just, even if we wanted to give them everything, we won't be able to. So we can develop these trials in a way in which they get something, but they don't get everything, because I don't think you can possibly give them everything. Even if you're talking about ozone, it might be cheap, but it does require, first, Dr. Richard has to be consulted. Second, you have to be the nurse to give it. It has to be given with a certain frequency. And I don't see you doing all that stuff at the same time. It's just impossible.
Right. Dr. Mole, are you on the line?
I am.
Okay. You want to talk something about the ozone. You want to talk about what you were telling me? Or?
I put in already about 8 consults, and I don't know if Dr. Richter's getting them because
he's right here. Go ahead, Dr.
Yes, I'm here, but I only was told about one and today.
we've put him in Palm Springs, and we've put him in Larkin, Maine. In the IC, every time I get an ICU patient, I try to consult you on, but we haven't been able to get it done.
Do you have his cell phone number?
No.
Franz, why don't you give me your cell phone number? Or you?
So I'm gonna type in the message.
Okay,
Perfect.
Louis, do you have access to the chat?
I'll get it.
All right, somebody give it from Dr. Mendes from the chat, please.
When you ask, what is it they tell you? The machine? What is it they told you? You said?
Yeah, the first time told me that they were having problems with the machine that they couldn't get done, and it was a patient that was in ICU bed 1.
Yeah,
the machine needed to get regulated. That's what I got.
No. It's at first there was a tubing issue. We got the tubing. I think we got everything now. So there's no reason why it can't be given now.
Yeah, everything's ready. The nurses are pretty much trained and everything. We just have to start doing it. That's all.
Hey, John, how did you feel after you got the ozone?
How do I feel? My arm was a little bit sore.
Yeah. Are you breathing better?
Purple and blue, but I mean, I didn't feel a difference because I wasn't sick at all. So.
Oh, okay.
Yeah, but no side effects or anything like that. I'm still alive. No air thrombi.
So we have 2 patients that are getting ozone, Dr. Richter, and also our resident John Din, chief resident. He also got it.
Exactly.
We can say we give it to ourselves before we give it to the patient. All right. Does anybody else have any comments or questions? Anybody? I think we're good then. We'll see you next Monday, same time.
Thank you.
Thank you. Thank you.
All right, thank you guys.
Thank you.
I'm having lunch, so if you guys want to join me.
Oh, yes, we heard that. Yeah.
We heard that, John.
Bye, guys.
Thank you very much.
Thank you.